Welcome to this tutorial video. So today we'll be looking at a topic that we call gametogenesis in anatomy, human anatomy. So before we even make progress, make sure that you subscribe to this YouTube channel, make sure that you like the video as well as share it. Now, today's topic is very important because we are looking at where you and I as human beings came from scientifically. So in as much as you can look at a human being complex, doing different things, you must understand that those human beings came from a single cell. And that single cell is what we call a gamete or a sex cell. So in today's session, we'll be looking at how do we form those gametes? How do we form those sex cells? Either female sex cells or male sex cells. So that process in which we biologically form those sex cells is what we call gametogenesis. It is important as somebody who is studying a healthy related course or program, you must understand this process. Because when you are talking about infertility challenges, they originate from this. If you are talking about reproductive health, if you are talking about, gene mutations, if you are talking about Down syndrome and all other different mutations that may take place in the reproductive life of somebody, you must understand gametogenesis. So to start with, if you look at this term, gametogenesis, it has got two terms or words in it. It has got a gamete here, which simply means a sex cell. Then genesis simply means formation. So gametogenesis is basically a process that involves the formation of sex cells or the formation of gametes. So you can see on your screen here to say these gametes that we're talking about can either be male gametes or female gametes. Now, a male gamete or a male sex cell is called a sperm. A female gamete or a female sex cell is called an ovum or oocyte. So we want to understand in this session to say, how do we form this sperm and how do we form this female gamete or ovum? And you know very well to say when these two fuse, okay, or when these two come into contact, there will be, of course, what we call fertilization. But before that, we want to understand to say, how do we form this sperm? How do we form this ovum? So that's what we will be looking at today. So just to give you a background understanding of the general topic, you must know these definitions. To start with, you are being told here to say gametogenesis is a biological process in which female and male gametes, or rather sex cells, are produced. A process in which female and male gametes are produced. That's gametogenesis. Now, there is a concept of meiosis here. Why are we talking about meiosis? We are talking about meiosis because gametogenesis is a process that involves meiosis. And I must emphasize that meiosis only takes place in sex cells. If you are talking about somatic cells, your muscle cells, those kind of cells do not involve meiosis. Meiosis only takes place in sex cells. And what we're talking about today are the very sex cells, the female sex cell and the male sex cell. Now, meiosis is a type of cell division in which occurs in gonads, right? In which there is reduction of the number of chromosomes from 2N or 46 chromosome to 1N or 23 chromosome. So what we simply mean is that in meiosis, remember when you looked at the cell cycle, you looked at meiosis and you realized that in that meiosis that you looked at, there was a reduction in the number of chromosomes. From 2N simply means diploid number of chromosomes or 46 chromosomes. you reduce those number of chromosomes to what we call a haploid number of chromosomes, or 1N. Simply means 23 chromosomes. No wonder we are calling it reduction. Okay? In some other books, you find that this meiosis is called reduction division. Why? It's because you are reducing the number of chromosomes from 46 chromosomes to 23 chromosomes. So if you find where they write 2N, it simply means diploid number of chromosomes, or 46 chromosomes. Where they write N or 1N, it simply means 23 chromosomes, or a haploid number of chromosomes. And I repeat to say, meiosis takes place only in sex-producing cells. And those are the gonads. All right. Now, you also come across this concept of what we call spermatogenesis. Remember from the introduction I said that gametogenesis involves the production of female and male sex cells. Now, if you are talking about males specifically, that process of forming gametes in males, we call it spermatogenesis, because we are producing sperms. No wonder we are saying that spermatogenesis is a biological process in which male gametes or sperms are what? Produced. But what about in females? Gametogenesis in female is called oogenesis. Now, it's a biological process in which female gametes or oocytes are produced. So you must familiarize yourself with this term oocyte. It's a scientific term for a female egg. So what did we look at this slide? We looked at gametogenesis being the overall process that involves female and male sex cell production. Meiosis, we have said, this is the type of cell division which occurs only in sex cells. And we have said that gametogenesis in males is called spermatogenesis because it's a biological process which involves the production of male gametes or a sperm. In females, we are calling it oogenesis, a biological process in which female gametes are produced. Now, this is a general overview of meiosis. Remember I said, if you are talking about a sex cell dividing or being produced, you cannot run away from meiosis because it is the only type of cell division in which you can produce sex cells. You cannot produce sex cells using mitosis. Why? You can see on top here to say, in meiosis, you reduce the number of chromosomes from 2N to what? To 1N. Meaning you are reducing the number of chromosomes from a diploid number of chromosomes to a haploid number of chromosomes, which is 1N. So the general concept behind meiosis, in case you didn't look at it, here is what happens. So you can see that you've got a primary cell in meiosis that you want to divide here. Now, since meiosis involves two processes, there is meiosis 1 and meiosis 2. So we will go through those stages for us to produce these sex cells that we're talking about. So in the first, cell, you can see that this is the primary cell that we are going to divide. So this primary cell, of course, it contains DNA content, okay? It contains chromosomes. So what happens is that we are reducing this primary cell from 2N using meiosis 1, then we form a secondary cell. From primary cell, we form a secondary cell. Now, you can see that from a primary cell, you've got 2N number of chromosomes. But when you form a secondary cell, you've got what? 1N. Meaning you have to know to say in meiosis 1, there's reduction in the number of chromosomes. They can ask you this question. It's a reduction division. But you will notice that from secondary cell, when you produce your final, daughter cells, okay, you are producing any number of chromosomes. That means that you are producing a haploid number of chromosomes. You can see from the top here to say 2N simply means 46 chromosomes. But what you have down here is 1N, or rather N, which simply means 23 chromosomes. And you can also see that in the first primary cell, you had one cell. But as you finish this process of meiosis, you've got 4 different cells. That is meiosis. Recap. In meiosis, you convert a primary cell from 2N to a secondary cell which has got any number of chromosomes. That simply means a haploid number of chromosomes. Take note that there is no change in the number of chromosomes. There is no change from the secondary cell to the final cell that you produce. The number of chromosomes is maintained. What you simply do is that you separate these sister chromatids into individual chromosomes. So in here you've got 23 chromosomes, 23 chromosomes, 23 chromosomes, 23 chromosomes. If this was a sperm, cell that you are trying to divide or to form, it simply means that here you've got this is going to be a final sperm cell, another sperm cell, another sperm cell, and another sperm cell. So that's why we're talking about meiosis, because it's the one that we use to form these gametes. Now, let's begin. Since we said that, we'll look at gametogenesis both in males and in females. So I've got Mr. John here, you can see, just trying to remind you to say this is spermatogenesis, meaning this is a process of forming gametes in males, and that is what we call gametogenesis. And you can see down here to say this is basically what? A sperm cell that we're trying to form. But before we look at this mature and potent sperm cell, let's understand to say how is it formed. And I must emphasize that the formation of sperm or sperm cells involves 3 phases. So this spermatogenesis, it involves 3 stages. You've got one, what we call spermacytogenesis. I'm going to talk about this. Then you've got meiosis. Then you've got spermiogenesis. So when you are talking about spermacytogenesis, we're talking about the primary, origin of the cell. The primary origin of the cell where this sperm cell comes from. Then meiosis, the very meiosis that I talked about, just trying to divide the number of chromosomes from 46 chromosomes to 23 chromosomes. Then spermiogenesis is a process in which this newly formed sperm matures. We're going to look at that. Note that this process of spermatogenesis takes place where? In the testis. Right? Specifically where? In the seminiferous tubules. So let's begin. Now, since we said that the process of spermatogenesis involves 3 stages, and the first one is spermacytogenesis. So you can see this word to say there is sperma, then cyto, meaning a cell. So you have got this word sperma, meaning a sperm, then cyto, a cell, genesis. So we're just trying to understand to say, where does this particular cell of the sperm come from? How is it formed? That's what we're looking at in the process of what? Spermacytogenesis. Now, we are saying that primordial germ cells, okay, which contain 46 number of chromosomes, or 2n if you want, okay, they originate from the walls of the yolk sac. So if you look at your embryology, science, you will know where during embryology, right, there is differentiation of, the newly formed zygote. It forms a blastomere, and it forms different, structures. And one of them is what we call the yolk sac. So it is from the yolk sac where you've got what we call these primordial germ cells, which contain 46 number of chromosomes, or 2n. Now we are saying that these primordial germ cells, okay, from the walls of this yolk sac that we talked about, arrive in the testis at 6 weeks, okay, and remain dominant until puberty. So somebody may be confused here to say, what does the sign mean at 6 weeks? So after fertilization has taken place, right, there's formation of a zygote. So now that zygote, when it's implanted in the female uterus, what happens is that there will be development. So after somebody is pregnant, okay, when the fetus reaches 6 weeks, this is why we're saying that at 6 weeks, the primordial germ cells from the walls of the yolk sac, they arrive in the testis at 6 weeks. Meaning somebody who's pregnant carrying a fetus, that fetus at 6 weeks, it's going to have the testis. It's why? Because this is where these primordial germ cells are going to arrive. I repeat, these primordial germ cells coming from the walls of the yolk sac, they are going to arrive at 6 weeks, okay, in the testis, and they will remain dormant until puberty. That is in males. So imagine this pregnant woman who is carrying a male fetus. That fetus now, when it reaches 6 weeks, these primordial germ cells are going to arrive in the testis of that fetus at 6 weeks, and they will remain dormant until puberty. I hope you've understood that part. So they will remain dormant until what? Puberty. Now, after puberty, you can see here to say after puberty, these primordial germ cells, they differentiate into what we call type A spermatogonia. They differentiate into what we call what? Type A spermatogonia. So after now this baby boy is born, then at puberty now, the body is going to remember to say, oh, at 6 weeks, when you were a fetus, these primordial germ cells, they arrived in your testes at 6 weeks. Now you've reached puberty, now you must start producing sperms because you've reached puberty. No wonder we are saying that at puberty now, the process continues where now these primordial germ cells, okay, these primordial germ cells, they differentiate or they change from primordial germ cells into what we call type A spermatogonia. And this type A spermatogonia has got 46 number of chromosomes, or what we call 2N, diploid number of chromosomes. The process continues. Now, this type A spermatogonia undergo mitosis to provide a continuous supply of stem cells throughout the reproductive life of the male. Remember that after a male reaches puberty, sperm production is continuous until they die. That is why this type A spermatogonia here, it will undergo mitosis. Remember, the overall idea behind mitosis is to produce a many number of cells. So imagine if you only had one type of spermatogonia, which is going to differentiate. But what a male needs is numerous number of sperms. You've been taught to say sperm count has got millions of sperms in it. And the only way the body makes that possible is by making sure that this type A spermatogonia undergoes mitosis. And I must emphasize, it's the only it's the only cell that undergoes mitosis. This type A spermatogonia undergoes mitosis to provide continuous number of sperms. So we've got a different number of sperms that are differentiating. So we are saying that some type A spermatogonia differentiate into what we call type B spermatogonia. And this type B spermatogonia still has 46 number of chromosomes, or 2N, which is the diploid number of chromosomes. Now, this type B spermatogonia now is the one that form what we call the primary spermatocyte. I repeat, we are saying that on this slide, you've got primordial germ cells that originate from the walls of the yolk sac and reach the testes at 6 weeks of the fetal life. And they are going to remain dormant until puberty. When a male reaches puberty, these primordial germ cells, they are going to wake up and say, let's continue. Then they will differentiate into what we call type A spermatogonia, which has got what? 46 number of chromosomes, or 2N. Then this type A spermatogonia is going to undergo mitosis to provide a different number of type A spermatogonia, which can now differentiate, as you can see here, into type B spermatogonia, which will then differentiate into what we call primary spermatocyte. Now, from this primary spermatocyte that we formed, we can now use this and make it undergo what we call meiosis, so that we can form those sperms or sperm cells that we want. Now, you can see very well here to say you've got meiosis here. This is the process of meiosis. So in this meiosis, you have got this now type A spermatogonia, right? That is differentiating. Let me show you this slide. Imagine now, this is your primary spermatocyte that you formed. So this one now must undergo meiosis 1 to form the secondary spermatocyte. This secondary spermatocyte must undergo meiosis 2 to form the spermatids, or the immature sperms. So imagine this was your primary spermatocyte. It undergoes meiosis 1. You reduce the number of chromosomes from 2N to 1N. So you form a secondary spermatocyte. This secondary spermatocyte undergoes meiosis 2 and form these spermatids, which have got 23 number of chromosomes on each spermatid. So that is what is involved basically in meiosis. So that's what you are seeing here. You've got a primary, spermatocyte. It undergoes meiosis 1. It reduces the number of chromosomes from 2N to N, and then it forms a secondary spermatocyte. This secondary spermatocyte, it undergoes meiosis too and form what? A spermatid. Okay, so these spermatids are going to be 4 of them: 1, 2, 3, and 4. Okay, that's what happens. So remember, we said spermatogenesis involves 3 phases. We've looked at spermatogenesis. We've looked at meiosis. Now we are going to look at the last stage, which we call spermiogenesis. So basically, what happens in spermiogenesis is that spermatid that you formed here after meiosis, remember, from primary spermatocyte you formed a secondary spermatocyte. From there you formed now a spermatid. 4 spermatids. 1 primary spermatocyte is equal to what? 4 spermatids. That is the rule of meiosis. Now, these spermatids are not mature. I mean, they're not mature. They can't fertilize an egg. They are not able to do that. So the body knows to say, okay, what do we do with them? We are going to make them undergo a process that we call spermiogenesis. This is a process that, these spermatids undergo series of morphological changes to form sperm, which has got what? 23 number of chromosomes, or 1N. So the whole idea behind spermiogenesis is that this sperm must mature. As a spermatid, it cannot fertilize an egg. So what the body will do now in this process of spermiogenesis, it will just make this sperm undergo morphological changes that are going to make it mature. You can see on your right here to say this is a full complete sperm which has got a nucleus, it has got the tail here, it has got the mid, okay, or the neck and the head. So there is now, in spermiogenesis, is just maturation of the sperm, which there is formation of the acrosome. You can see that the acrosome was formed here. Condensation of the nucleus. You can see that this nucleus has what? Condensed, which contain the genetic material. There is formation of the head, neck, and tail. You can see that there is the head here, there is the neck here, and there is the tail. Now, you must know to say the total time of sperm formation from spermatogonia to spermatozoa is about what? 64 days. So every sperm that is alive today was formed 64 days ago. That you must know, because they can ask you. So this is the part that concludes, spermatogenesis in males. Why? It's because we said spermatogenesis in males involves 3 stages. We've looked at spermacytogenesis, where you had the primordial germ cells, originating from the walls of the yolk sac and arriving in the testis at week 6 of fetal life, and they remained dormant until puberty. That was the part which we talked about, spermatogenesis. Then in meiosis now, we talked about the primary spermatocyte that was formed from what? Type B spermatogonia, which had to undergo what? Meiosis. From primary spermatocyte to secondary spermatocyte until it formed the spermatid. Then we looked at now spermiogenesis. We said this is basically a process that involves maturation of the sperm, making it to have the head, the neck, and the tail. So this is how now the male sperm is formed. So today you've known to say this sperm that was formed, or this sperm that is alive today, was formed 64 days ago. That is the overall idea. Now, somebody is asking to say, what about females? Don't worry. We're also going to talk about sex cells formation in what? Females. And that process, we call it oogenesis. Oogenesis, from the word oocyte. So you can see that we've got a female here and we want to form this ovum. So let's begin. Now, oogenesis is similar to spermatogenesis, but there are some differences, and they like, asking you questions on these differences because they want to know if you understand the difference between gamete formation in males and gamete formation in females. So, how does oogenesis start? It starts from here. The very primordial germ cells, okay? Primordial germ cells containing 46 number of chromosomes, or 2N, from the walls of the yolk sac, they arrive in the ovary. Remember, this is what? This is now a female fetus that we're talking about. A female fetus. So that woman is pregnant, who is carrying a female fetus. At 6 weeks, you've got these primordial germ cells, okay, from the walls of the yolk sac that arrive in the ovary at 6 weeks, and these, they differentiate into oogonia. Remember, in males, they remain dormant until puberty, but in females, when they arrive in the ovaries at 6 weeks, they differentiate into what we call oogonia. And this oogonia has got 46 number of chromosomes, or 2N. Now, which populate the ovary through mitotic division. So this oogonia, it also now undergo mitosis just to form many number of oogonia. Okay? So it's going to undergo mitosis. What happens after that? Now, we are saying that oogonia, okay, enter meiosis 1 and undergo DNA replication to form primary oocyte. If you remember in males, when the primordial germ cells remain dormant until puberty, okay, they remain dormant until puberty. But in females, before even the female, baby is born, you can see to say meiosis 1 has already started. So these are the questions that they ask you. Oogonia will enter meiosis 1. You can see that this is even before birth, and undergo DNA replication to form primary spermatocyte, primary oocyte. In males, the primary spermatocyte was formed after puberty, but in females, you are forming this primary oocyte even before birth. And we are saying that this primary oocyte has got what? 46 number of chromosomes, or 2N. This is supposed to be 2N, not 4N, 2N. Now, all primary oocytes are formed by month 5 of fetal life, meaning before the female baby is born, it has formed all its primary oocytes. No wonder they like saying in science to say female eggs are older than the actual person. Why? It's because they were formed at month 5 before even the fetus is born. So oogonia enter meiosis 1 and undergo DNA replication to form primary oocyte, which has got 46 number of chromosomes. All primary oocytes are formed by month 5 of fetal life. So that woman who is pregnant, in month 5, the fetus, the female fetus that she's carrying, it has already formed its primary oocytes. No oogonia are present at birth. No oogonia are present at what? Birth. Remember, in males, primordial germ cells, they had to resume the process of spermatogenesis, and they had to form spermatogonia. So in that case, spermatogonia was present after birth. But in females, there is no oogonia at birth. Why? It's because this oogonia will undergo different DNA replication to form primary oocyte. So no oogonia is present at what? Birth. You continue. Now, the primary oocyte now, remember, from here we said oogonia differentiated to what? Primary oocyte. So this primary oocyte now is going to remain dormant in prophase 1 of meiosis 1. This you must know. Remember, meiosis has got 2 phases, meiosis 1 and meiosis 2. Now, this primary oocyte now will remain dormant in prophase 1 of meiosis 1 from month 5 of fetal life until puberty. So this is the stage where this female, primary oocyte is kind of blocked or goes to rest. It will go to rest in prophase 1 of meiosis 1 at month 5 of fetal life, and it's going to wait until this female baby reaches puberty. Now, after puberty now, we are saying that about 5 to 15 primary oocytes, those that remain dormant now at month 5, they're going to wake up. Now, about 5 to 15 of them will begin maturation with each ovarian cycle. That ovarian cycle, the menstrual cycle that we talk about 5 to 15 of them, the primary oocytes, they'll start being mature with each ovarian cycle, with usually only one reaching maturity in each cycle. Remember, a woman under normal circumstances must ovulate only one egg. That's why we are saying that in as much as 15 or 5 of them may begin maturation, but only one of them is going to reach full maturity in each cycle. So every menstrual cycle or ovarian cycle that a woman undergoes, about 5 to 15 primary oocytes are going to start that process, but only one of them reaches the final mark, reaches the final line. That you must know. Now, remember that during the ovarian cycle, okay, when there is formation of those those ovums or eggs. During the ovarian cycle, and triggered by luteinizing hormone, says a primary oocyte completes meiosis 1 to form two daughter cells. That is the secondary oocyte, which has got 23 number of chromosomes and the first polar body. Remember, I said that in meiosis, when you've got a primary cell, that primary cell undergoes meiosis 1 to form a secondary cell. And it is on that particular stage from meiosis 1 where you're reducing the number of chromosomes from 46. So meaning these primary oocytes, this primary oocyte that we're talking about, it's going to have 2n number of chromosomes. But when you form this secondary oocyte, it has got 23 number of chromosomes or 1n. And you also form the first polar body. You form the first polar body. So you must know that, remember, in males, one primary oocyte formed 4 spermatids, but in females, when you start with one primary, in males, one primary spermatocyte formed 4 spermatids, but in females, if you've got one primary oocyte, it's only going to form one secondary oocyte. Why? It's because the other 3, they differentiate into what we call polar bodies. But when this primary oocyte completes meiosis 1, it forms the first secondary oocyte and the first polar body. We continue. Now, this secondary oocyte, promptly begins meiosis 2. It's going to begin meiosis 2. But it is arrested in metaphase of meiosis 2. So, in meiosis 2 of metaphase 2, this secondary oocyte is arrested again. So remember, the female oocyte is arrested twice. The primary oocyte was arrested at what? Prophase 1 of meiosis 1. Again, here in the secondary meiosis, it's arrested at what? Metaphase 2 of meiosis 2, about 3 hours before ovulation. Now, the secondary oocyte is going to remain arrested in metaphase 2 of meiosis 2 until fertilization occurs. So you must know to say that egg, or that ovum that a woman ovulates, it has not finished meiosis 2. Why? It's because it is arrested at metaphase 2 of meiosis 2, and it's going to remain dormant until fertilization occurs. That's when it's going to complete its meiosis 2. Now, when fertilization occurs, the secondary oocyte completes meiosis 2. So, meaning that the secondary oocyte, that same egg that a woman ovulates, it has not completed its meiosis 2. The only way it's going to do that is when it's fertilized. So every time in the menstrual ovarian cycle, a woman ovulates, and that egg that ovulates, it has not completed the meiosis 2. It has not finished the process. Until that particular day that woman is going to be impregnated, or where fertilization is going to take place, that is when now it's going to complete meiosis 2. Alright. So this is the overview that we're talking about. So you had oogonia here. Remember, when oogonia undergoes mitosis to form different types of oogonia, this oogonia differentiates into what? The primary oocyte. This primary oocyte undergoes meiosis 1, you can see. If it undergoes meiosis 1, it forms the secondary oocyte. You can see here to say there is a reduction in the number of chromosomes. The primary oocyte has got what? 2n for the 6 chromosomes. But the secondary oocyte has got what? Any number of chromosomes. That is, means haploid number of chromosomes, or 23 chromosomes. Then this secondary oocyte now will eventually form one. This secondary oocyte now will eventually form one secondary oocyte. This oocyte that we're talking about is going to be arrested where? At metaphase 2 of meiosis 2. You can see here to say at metaphase 2 of meiosis 2, the secondary oocyte is going to be arrested, and the only way it can complete that process is after fertilization. So that's what we're talking about here. But you can see to say from one primary oocyte, you only form one ovum. The rest of them are polar bodies. In male spermatogenesis, you saw to say you have got one, primary spermatocyte here, but you were able to form 4 spermatids. But in females, you're only forming 1. I said in as much as 15 or 5 of them may start, but only one of them is going to reach full maturity. So where do the rest go? They just become what we call polar bodies. These polar bodies, after some time, they just degenerate and disappear. So that is what happens when you're forming female gametes, or when you're forming a female sex cell or a female egg. So these are just some questions that you can go through to understand if at all you've understood those questions. So make sure that you subscribe to this YouTube channel, you share the video, and watch out for other upcoming videos. Thank you so much.